Hey, I'm Mr. Williamson. This is about reading DNA. We've already talked about the structure of DNA, the history, what Watson and Crick need to do. It need to be able to store information, to be able to copy itself with precision, need to be able to make mistakes, and need to be able to be read. They knew about, we talked about the structure. Adenine is a purine that has a double rings and double bonds. It pairs up with thymine, guanine, and cytosine pair up. You should know all that. If you don't, probably should attend class because we're going to talk about it. All right. We've talked about how it can copy itself. DNA helicase. You don't need to know that. DNA helicase, an enzyme, comes in, unzips the DNA, and DNA polymerase comes in and pairs it up to the template. T's with A's, G's with C's. We talked about how to make mistakes. Sometimes an A and a G combine and it's not corrected. Most of the time it is corrected, but sometimes it's not. And that can be a problem or maybe not, depending on where it is in the DNA. If we're talking about a skin cell and you damage the liver cell portion of the DNA, nothing happens. It's again like the example I used in the last video. If we're on chapter 12 and your brother destroys chapter 5, you don't care other than you have to pay for the book, right? Okay, so the question is, though, how do we read DNA? That's the question. What do we need to do to read DNA? How does the cell read DNA to get a protein, the code for a protein? Well, let's take a break before we get started here, all right? How many words can you make out of the following 4 letters? Now, the class record is 36. We're not going to go into it. And honestly, you don't need to do this. I'm just trying to show you something. If you'd like to, by all means, take the time to do it. Don't do it. It's not important. There's your 4 letters, A, T, R, and C. All right, right about now, I'd be playing Jeopardy and giving you as much time, Jeopardy music, and giving you as much time to do this as you want. Don't waste your time, all right? So how many did you come up with? Assuming that you really did it, how many did you come up with? How many of those were 3-letter words? Okay, let's talk about making a math question. How many letters, how many combinations of 3-letter words can you make out of the 4 letters? All right, it doesn't matter if it makes sense or if it's actually a word. I'm just asking how many combinations of 3 letters can you make out of those 4 letters? All right, well, it's simple math. It's 4 to the 3rd. All right, you have 4 letters, and you're going to put them in combinations of 3. That's the formula is 4 to the 3rd. So 4 times 4 is 16, times 4 is 64. So there are 64 combinations of 4 letters if we only take them in combinations of 3. All right, what if we substitute in the bases? What if we put in the bases of DNA? A, C, G, and T. Well, still 64, 4 letters, and we're taking them in combinations of 3. The way you need to think about this, the bases, A, C, G, and T, are the alphabet for DNA, and eventually when we get farther on, for RNA. The bases are the alphabet, all right? If you have an alphabet, you have to have words. All of the words in DNA are 3 letters long. That's the maximum. This is how the DNA reads, or the cell reads the DNA, in 3 base combinations. Each combination is called a codon. All right, a codon is the code for 1 amino acid. So think of the bases as the alphabet. Think of a codon as the word for DNA. All right, so you've got a language. We have a language. We have 26 letters in our alphabet. We can put them in combinations of any length, anywhere from 1 letter to I don't know how many. All right, look that one up. How long is the longest word in the English language? But my point is, we have 26 letters in the alphabet, and depending on what order we put them in, it changes the meaning of the word. For instance, if I write form almost neatly, that's a little different than if I write from. Okay? 2 different, same line, same word, same letters, different words, just on the order of just reversing 2 of them. Okay? So you have We have DNA has an alphabet, except that it's only not 26 like ours. It's only 4 letters, and it has a vocabulary. It has words that are only 3 letters long. You can't put them in unlimited numbers. You have to. Every word in the DNA language is 3 letters long. It's called a codon. All right. There is another nucleic acid. We've been talking about DNA all this time. There's another one. It's called RNA, ribonucleic acid. All right. RNA is very similar to DNA, except it's a different sugar. Instead of deoxyribose, it has ribose. So that's why it's ribonucleic acid. Instead of double strand, so instead of a double strand, you have like that, and you'd have the base pairs. You only have 1 strand, one side. RNA only is a single side. And instead of thymine, we're using uracil. So while DNA has A and T and G and C, RNA, probably should put this up here for you. RNA has A and U, G and C. Doesn't have the T. These two are replaced. Uracil replaces thymine. You should know that. You need to know that. So there are 3 types of RNA. mRNA, which stands for messenger RNA. tRNA stands for transfer RNA. And rRNA, which stands for ribosomal RNA. Now, this is the end of the video. What we need to talk about is how do we read the DNA? We've talked about the alphabet, T, C, G, A. We've talked about the words, codons, every combination of 3. There are only 64 words in the entire language of DNA because it only has 4 letters. So what we need to talk about is how do we read it? How do we get it to become a protein? That's the role of messenger RNA and transfer RNA, and a little bit of rRNA. That's what we're going to talk about in the next video. All right? So again, let's take a real quick review. What we talked about in this video was the codons, the alphabet, A, T, G, and C of DNA, the words, which are only 3 letters long, called codons. There's only 64 of them. And we learned about a new nucleic acid, RNA. RNA is single strand. It replaces thymine with uracil. So there is no thymine in RNA. There is uracil, which, by the way, makes a great test question. If you see a strand of letters, let's just say C, it's a C. Is that RNA or DNA? It has to be DNA because it has T's in it. On the other hand, if you were to see, for instance, is that RNA or DNA? Well, because there's U's, it has to be RNA. You can tell if it's RNA or DNA. If it has a T, it's DNA. If it has a U, it's RNA. It makes a good test question. But like I said, we need to talk about how we do the whole process of protein synthesis using these codons in the DNA and essentially, eventually, the RNA. You need to know about the 3 types of RNA: mRNA, tRNA, and rRNA. Hopefully you took good notes. If you have any questions, ask them at Edpuzzle or ask me in the classroom. Good luck.